All right, let me get my pointer started early here. Boom. All right, so we're going to talk about syncytial specification. So here's your nonsense picture. Maybe, um, maybe you can make sense of it after we talk about what it is, because I see like maybe this could be like multiple nuclei. Um, we'll see. Okay. So, oh, spacing on this is all messed up, but so syncytial specification is where there's a cytoplasm that contain many nuclei called syncytium. So, specification of these cells is called syncytial specification. So an example of this would be Drosophila melanogaster, or the fruit fly, which we do a lot of developmental biology with. And so there's like 13 nuclear divisions before any cytoplasmic cleavage occurs. So here's kind of an example. So here is like a Drosophila's egg. So there's these nuclear cycles during Drosophila's early development. And you can see that we have one cell with a bunch of these little red nuclei. And this is called the pre-blastoderm cycles, 1 through 9, where you just get this nuclei that's increasing more and more and more. And so you make all these nuclei, and then they all go line up around the edge of the cell. Well, not all of them, but a lot of them go line up along the edge of the cell. And then you have these cell walls form around them. So instead of developing each cell splitting in half multiple times, it just starts by replicating all these nuclei and then puts them along the edge of the cell, and then builds cell walls around them to become the new cells. So, and then there's an interaction between these nuclei and cytoplasmic determinants. So this is kind of shown here, some of these stained morphogenic factors and the nuclei here. Those determine the cell fate. So what, instead of your location or whatever, it's a combination of the nuclei and cytoplasmic factors. So it's kind of a combination of both conditional and autonomous. Okay, let's see. So this is, there are these microtubules that are like actin and microtubule interacting proteins and centrosomes that hold these nuclei in place and pull them to the edge of the cell. So you can see kind of the nuclei here and all these little fibers coming off of them. And so that prevents them from drifting off and helps to sequester them to the edge of the cell. And so, like I said, we're going to talk about gradients coming up and like differential expression of like Hox genes and things of that nature. But where you are based on these gradients determines what your cell fate's going to be. So you can see that like this GFP staining on this actual Drosophila egg here, very bright on this end, very dim on this end. So if we were to graph that, you could see that on this anterior end, so towards the head, you can see that there's a lot of whatever this protein is. So say hunchback or whatever, morphogenic, or it's probably bicoid is the name of the morphogenic factor. So it's really high on this end, and then it gradually goes down to the posterior end, right? And so based on your presence of these morphogenic factors and how much of each of there is, that's what determines what your fate is going to be, right? So in this case, we have bicoid, which is in red here, which is really high on one end, and then it why can't I think of the word? Disperses slowly across the cell, and the concentration becomes much less at the posterior end. And then conversely, there's gradients going in all three directions, right, or all three dimensions. And so here we have a gradient for caudal, which is another morphogenic protein that is very high at the posterior end and very low in the anterior. So if you're a nuclei and you're in an area that has high bicoid but very low caudal, like no caudal, then you know that you're supposed to be the anterior or the head of this Drosophila. If you're somebody that has a nuclei or a cell that divides, gets sequestered this nuclei, that has high caudal and very low bicoid, then you know you're going to be the posterior or the tail end of this, right? And then if you're somebody in the middle, say you have equal amounts of bicoid and caudal, then you might be something like a wing or something that's like in the middle of the fly, right? So all these gradients, we're going to talk more about these gradients in the Hox genes and these developmental genes. They are what help to differentiate in this case of syncytial specification. So these are just, this is a quick, another roundup provided by the textbook of the three types. We already had one that compared conditional and autonomous. So this is just for your information. I'm not going to rehash it. I'll just add that syncytial predominates most in insects. Specification of body region by interaction between the cytoplasmic regions prior to cellulization of the blastoderm. So we have these gradients before the cellulization. No rigid sulfates. And then after cellularization, like I said, it's kind of a combination of autonomous and conditional specification are seen. So that is it for specification. So we're done with that. I believe at the beginning, the next unit, we're going to talk about these gradients and how these gradients are formed before we get into gastrulation, because this is all kind of predominantly happening at these early cell stages. And so we want to kind of get those mechanisms down for how this differentiation happens, because that's important for gastrulation and organogenesis and things like that. So thanks for watching, and I will catch you in the next one.